So this video will take you through the limiting factors for photosynthesis. But before we start doing that, I'd like you to recap the light-dependent and light-independent reactions. So could I ask you to pause the video and copy the diagram, and then complete the diagram with as much annotation as you can to show how ATP is produced during the light-dependent reactions. Okay, so you should have completed the task. So now you've got a fully annotated diagram, and I want to take you through the things that you should have added to your diagram. So first of all, I would expect some sort of representation here of light energy striking the chlorophyll. And the important bit here is that we've got light energy. We've got the energy word in there, and the fact that it's being absorbed by the chlorophyll molecule. And the chlorophyll molecule is within the within the photosystem, which actually is the second photosystem, but that doesn't matter. So it's within the photosystem, so it's one molecule of many embedded there in the thylakoid membrane. Okay, so you may even have labelled up your thylakoid membrane. And that you might have labelled up also, and if you haven't, you can add this now, that this is inside the thylakoid disc. Inside the disc space, if you like, at the centre of the disc. So this is the thylakoid membrane itself, and that means that this is the stroma, this is outside of the disc. Okay, so light energy is absorbed by the chlorophyll molecule. This causes the electron to be excited and raised to a higher energy level. So that's kind of represented, so it isn't represented in this diagram. What's represented is what happens as a consequence, which is that the electrons are lost from the chlorophyll, and you may have called that an oxidation. Okay, but the name of that process when light energy is absorbed by the chlorophyll and it loses electrons, excited electrons, is photoionization. So you may have annotated there, see whether I can fit it in, photoionization, okay, which is of course the key word you want to have in there to describe that process. Okay, so now what happens to the electrons? Well, they move down the electron transport chain by a series of redox reactions. Okay, so you may have labelled up these purple blobs as electron transport proteins. So as the electrons move down the electron transport chain, they move down energy levels, okay, and that energy is actually utilised to pump protons across the thylakoid membrane into the thylakoid disc space. So we get a build-up of protons inside the disc. Okay. Now, the chlorophyll needs to replace those electrons so this process can continue. Okay, so you can see here, the top of the diagram, inside the disc, we've got a process happening which allows us to do that. So here we've got water splitting, and we've got our products. So we've got electrons, protons, and oxygen. Okay, you can, if you want to, double all those up so you can make a full oxygen molecule. That's our waste product from this process of light-dependent reactions. And that process you would have labelled up as photolysis or photolysis, if you like. Okay. So now we've just got to make our ATP. So we've got our proton gradient that we've established. So we've got lots of protons inside the thylakoid disc space and a channel for them to move through, which is in our ATP synthase. So we show then that our protons will move back down, back across the thylakoid membrane, down a concentration gradient this time. And as they do that, the enzyme ATP synthase catalyzes a condensation reaction between ADP and phosphate, and we get ATP produced. Okay. Super. I can't think of anything else we would add. So that would give you your sort of 4-mark description of how ATP is produced during light-dependent reactions. Sometimes, of course, they'll ask us to concentrate on one process. So obviously, if they ask us about photolysis and how that is used, as we did in class, in making ATP, then you'll get that process will be worth a mark point, but we'll still have to cover the whole of this. So whenever we're asked about producing ATP during these reactions, it's everything that we've got to mention. If we're asked about photoionization, then there may be more mark points around the process itself, but we'll still have to do the whole of this process if we're asked about how ATP is produced during light-dependent reactions. So no matter where they shine the spotlight in this process, we do need to give them the whole process. Okay, so what about our light-independent reactions? So I'm going to ask you to do a little bit of AO2 here. So can I ask you to sketch the graph? Okay, and then note down the trends and then explain these. Okay, so you should have unpaused the video having done the task now. So the first thing I would do if I were answering this question in an exam is just a quick sort of reminder to myself of Calvin's cycle of the light-independent reactions. Remember, we can refer to the light-independent reactions as Calvin's cycle after the scientist that discovered them, using his lollipop experiment. So get my basics in. So I've got my products of the light dependent reactions here. My reduced NADP and my ATP, and they're used obviously in that reduction step of GP to TP. ATP provides the energy and NADPH provides the hydrogen in that reduction step. Remember, we have 2 GPs and 2 TPs for every VBP. And we do require ATP to phosphorylate TPs as part of the resynthesis of our RuBP. So that's why there's no phosphate coming out on this arrow here. So that's a summary of my Calvin cycle. Okay, so let's explain our trend, or we'll describe our trends first. So in the light, at first, there is a sort of time delay. There is a time delay as the radioactive carbon dioxide is added before all of the GP and the RuBP become radioactive. But once they do, then the trend is that the relative amounts remain the same. Yeah, so that would tell you that GP is being produced at the same rate as RuBP is being produced and used. So all the reactions are taking place at the same rate. As RuBP is being broken down to GP, it's then being recycled to RuBP at the same rate. Okay. So the proportion of those two molecules remains the same when the light is on, because those reactions are going at the same rate. However, when we turn the light out, there are some changes to what is available in the chloroplast. So when we turn the light out, the light dependent reactions stop, and so these products here are no longer produced. So we don't produce reduced NADP and we don't produce ATP. So without them, the reduction step of GP to TP does not happen. And if we don't produce TP, then we will not regenerate our RuBP. And that explains why the GP is accumulating, it's going up, and it's starting to slow down, that, so we'll think about that as well. And our RuBP is dropping, so that's our general trend. Okay, so RuBP is being used up because the carbon fixation continues, so this reaction here continues, but the reduction step to TP stops. So RuBP is being used up and converted into GP. But GP is not being converted into RuBP, so we're not making any more RuBP, it's being used up. Once the RuBP is completely used up, then we can't form any more GP, which is why it's starting to slow down here at the top of the graph. Okay, so today's video then is about the limiting factors of photosynthesis. So, could I ask you, first of all, what is a limiting factor? And what factors do you think limit the rate that photosynthesis happens and why? Okay, so I want you to just stop the video here and to write some ideas down. So what are limiting factors and which factors do you think limit the rate of photosynthesis? Okay, so now we should be joining me back. Okay, so what's a limiting factor? Okay, so what's this video about? Well, a limiting factor is a factor that limits the rate of a reaction. When you're looking at data sets, you can spot a limiting factor because if you increase that factor, you will get an increased return in rate. So it is that factor that is limiting. So we've met this before. We met this in our enzyme topic in year 12. So we talked about substrate concentration limiting the rate of reaction. So let me just remind you what that looked like. So those graphs, enzyme or substrate concentration on the bottom, and then rate of reaction of an enzyme controlled reaction on the y-axis, and we would go like this and then it would level off. Okay, so what we would say is, this was an enzyme concentration graph, is during this stage of the graph here, where as you increase the enzyme concentration you get an increase in the rate of reaction, we would identify that at any point on that line the enzyme concentration is the thing that's limiting the rate of reaction, because the evidence is when you increase it, you do get an increase in rate of reaction. But as you'll remember from these graphs, they do level off. Okay, and at the point where they level off, it's no longer the enzyme concentration that's limiting the rate of reaction, because as we're increasing the enzyme concentration, we're not getting a return in terms of rate of reaction. So, from data sets then, we've already identified limiting factors, and we're going to be doing that again in the video today. So we've met that term limiting factor, so a factor that inhibits the rate of a reaction. We know with a data set that we can identify that in data, because if it is the limiting factor and you increase it, you will get an increase in the rate of that reaction. So you'll be able to find what's limiting at a particular time. So what about photosynthesis then? What about the factors that limit the rate of photosynthesis? Okay, so what have you got in your list? Have you got light? Okay, I'm hoping you have light, because obviously our light dependent reactions depend on light being absorbed by our chlorophyll, which in turn can excite our electrons. So it's a physical reaction. We've talked about that in class, but one that if you don't have the light, then it won't happen. Okay, so it's going to be a fundamental limiting factor to the rate of photosynthesis. So we tend to write it as light intensity. Okay, And in practicals, we can change this by moving a lamp closer to or further away from the sample during the experiment. Okay, so we've got light intensity and hopefully a Y identifying that it's required for photoionization to happen. Without photoionization, nothing else would happen in photosynthesis. Okay, but that's not the only limiting factor. So what about the other things that you know would limit the rate of reaction? Well, we've got, hopefully, carbon dioxide listed as one of the substrates, okay, and temperature, because in biology we know, particularly for our Calvin cycle now from last lesson, that if we've got enzyme catalyzed reactions, and of course we've got a named enzyme in Calvin cycle, which is rubisco, those reactions speed up at first as you increase temperature to the optimum, but then above optimum as you increase temperature, that factor, temperature, would cause proteins to denature and would start to reduce your rate. So, temperature up to optimum, why would it limit the rate of reaction? So you should be able to link to kinetic energy and collisions between the enzymes involved in the light independent reactions and the substrates. Okay. And you've got rubisco, which you can talk about specifically. So if you've got a very low temperature, then what will happen is we will have a few collisions between our RuBP and our carbon dioxide and our rubisco enzyme active sites, and so that reaction will be slow, and if that reaction is slow, that will slow the whole of Calvin cycle. Okay, right, so final one then is our substrate here. So again, in our reactions of Calvin cycle, RuBP reacts with carbon dioxide, so it's a substrate. If you've got a limited concentration of carbon dioxide, that would again limit the number of enzyme substrate complexes, because now it's a substrate concentration limiting the rate of reaction. And again, that would reduce the rate at which we produce GP. So every single one of these you would need to be able to take to that kind of level of detail in your descriptions, and we'll have another go at that in a minute. Now, one of the other substrates that you might have thought about was water. Okay, now that's never a limiting factor for photosynthesis. I know that feels wrong because you know you need it for the process of photolysis or photolysis, but think about what you know already about gas exchange in plants. Okay, so if there is a high light intensity and photosynthesis is taking place, then the stomata are open to allow carbon dioxide to enter the cell for this process. If the water content of the plant reduces, then we know transpiration, loss of water from the leaf, would be reduced by the plant by closing the stomata, and as soon as you close the stomata, the limiting factor would be carbon dioxide. So we never get to a point where water is a limiting factor, because the impact of less water for the plant causes carbon dioxide to become a limiting factor because we close the stomata. Now the examiner loves that because that's real synoptic thinking on your part. Okay, so What we want to do is to just lift your notes now to include this kind of detail, because they are just bog sort of standard notes, because this is all application of knowledge, okay? And you might want to record some of that in your keynotes. Okay, so let's think about this data set here. You've got a similar graph in your keynotes. One of the things I'm going to ask you to do first of all is to look at this data set, okay, maybe sketch it down so that you can annotate ideas on it. And first thing is, can you identify on the graph somewhere where the light intensity is the limiting factor? Okay, so where on this graph is light intensity a limiting factor? Put a nice big red cross where that is. Okay. Right, so have you had a go at that? So have you identified where you think light is limiting? Well, on this graph, our x-axis is light intensity. So as you're increasing light intensity, you can see at first, in the first part of the graph for all the lines, the rate increases. So it's fine to place a cross, to say a red cross, anywhere where the line is still increasing. So all these would be correct. Okay. Obviously, eventually, light is no longer the limiting factor, so they could be asked that. So, for example, in our first two lines here, when we reach 4 arbitrary units, you can see that the line levels off. So it doesn't matter that we're increasing light intensity, something else has become a limiting factor. Okay, so then we've got to start to think about, at the point where light is no longer a limiting factor, what is limiting the rate of reaction? Okay, so this is where we can see the other factors that have been investigated in this graph. We can see that we've got an experiment where the carbon dioxide concentration is low, and we've changed the temperature. And then we've got one at the top, or experiments at the top here, where the carbon dioxide concentration is high. Okay, and we've changed the temperature. So we can make comparisons between these lines to say how much each factor is limiting at each light intensity. So for example, if the examiner said at an arbitrary unit of 5, arbitrary unit, so if the light intensity is 5 arbitrary units, how much is the carbon dioxide limiting the reaction, and how much is temperature limiting the reaction? Which of the two is limiting it most? So you could do a comparison then between your two experiments. Okay, so where you've got a low carbon dioxide concentration, you can see that we've only got a small return as we increase the temperature 10 degrees in rate. You can be able to quantify that. Whereas at a high concentration of carbon dioxide, the return in increasing your temperature is much bigger. Okay, and then if we do a comparison between the same temperature and increasing your carbon dioxide concentration, you can see that we've got a massive increase in rate of reaction at both temperatures. So you can quantify by how much the reaction was limited by each of these factors. You could be asked to extract that data from the graph. So it's just getting used to reading graphs and understanding that you've got these factor graphs where you can be interpreting the impact of 3 different limiting factors at the same time. Okay, So what we want to do now is, once they've got you to extract data to say how much a factor is particularly limiting photosynthesis under certain conditions, or to identify when light is limiting within X, then they usually go on to ask you to say why that factor is limiting. So for each of your factors, you need a good biological explanation. Now in your keynotes, I haven't put down the detail because it's application of knowledge. So you're applying your knowledge of the light dependent and light independent reactions now to explain these limiting factors. So you don't need to learn it off by heart. You should be able to just apply your knowledge. But let's see, you might want to make some additional notes in your key notes. Right, so now I want you to take the photosynthesis limiting factors AO2 challenge. So I want you to use all of your biological knowledge and try to give detailed responses to each of the following. So I want you to imagine that you've had a look at one of those multi-factor graphs, and then the examiner has asked you to explain one of the trends. So there are 4 trends that you could possibly be explaining. So why is the light intensity limiting the rate of photosynthesis? Why is the temperature limiting the rate of photosynthesis? Now that's at a low temperature, but also if your data shows it's a high temperature, it would change your answer. So I want you to do that one too. And then finally, why is carbon dioxide limiting the rate of photosynthesis? So we would be able to give a good explanation for any of those trends. Okay, so pause the video and give it a go. And then I'm going to review with you. Okay, so let's have a look at this student response and see how they've done. Okay, so they've said if the light intensity is limiting, they're going to say there's less light energy to be absorbed by the chlorophyll, so fewer electrons excited and moved down the electron transport chain, so it's going to be less reduced NADP and ATP produced, limiting the rate of the light independent reactions. So the only advice I would give to them is make sure that you write out light independent reactions here. But other than that, there's good depth here, isn't there? This is the kind of detail that we want. So you've got to think about how they're going to design a year 2 mark scheme for this. So it could be that they've got less energy absorbed by the chlorophyll as a mark point, less light energy absorbed by the chlorophyll, and less products of the light dependent reactions as the mark point. So by putting that lovely detail in that this student has, they should get the 2 marks for that detail. Okay. So it's making sure that you put enough detail in your response that no matter where the mark points are, because the mark points will be for detail, that we give a good answer. Okay. In contrast to this, a poor answer would be to sort of try to summarise all this in a line, okay? So to not put the detail in. So if you haven't got all the depth of the light dependent reactions in your answer, add it now and make sure it's in your keynotes if you have to learn it off by heart. If this just happens naturally, because this is you explaining, it's AO2, it's not something really you should have to learn off by heart, you know all this stuff already. Then you don't need to annotate your notes. But if you feel you need to make little flashcards of these, don't worry, do that. At least then you'll be definitely getting the detail. Okay, the next one then we're going to look at is temperature at the bottom of this slide here. So why is temperature limiting the rate of photosynthesis? Well, now we've got to go to our enzyme knowledge from year 1. So let's see what this person's put. Less kinetic energy, so less collisions between rubisco and carbon dioxide and RuBP. So instead of just doing generic enzymes, they have been specific to the enzymes involved in the light independent reactions. So fewer enzyme substrate complexes form. That would mean there's less GP produced, less TP produced, less RuBP recycled. So thinking about how this might be marked, it may be that just one of those is the mark point. It's that depth. So the idea that they've put all of them, given all the detail, it's taken a little bit longer. And it's likely to be that less kinetic energy is going to be a mark point, definitely. But to answer it fully, we would have to link that to collisions. So I think they've done enough there to get their 2 marks as well. Maybe you could have put a little bit more, but I think that should just about do it for 2 marks. Okay, because I've answered them in a different order, I'm now going to review the carbon dioxide one before I do the high temperature. So why is carbon dioxide limiting the rate of photosynthesis? So this student has said, well, carbon dioxide reacts with RuBP to form GP in the light independent reactions. If there is less carbon dioxide, there'll be less GP produced, and so less GP will be reduced to TP, resulting in a slower production of glucose and recycling of RuBP. So again, they've not just said it's a substrate and substrates limiting the rate of reaction. They have gone into detail about how the reactions will be limited and what products would be reduced. Okay. The only thing that you might want to add in there is that the idea it is a substrate might be there in the mark scheme as well. Okay, but I think there's enough there to get you 2 marks. And then finally, if you were doing a data set and temperature was limiting, but when you looked at the temperature, you weren't doing low temperatures like 20 and 30 degrees, but something that you know was a very high temperature, then okay, and finally, why is temperature limiting if it's a high temperature? So say you're looking at the graph and the temperature is the limiting factor, but it's a very high temperature, then your explanation would be a little bit different. So if it was a high temperature, you'd be using your knowledge from year 1 enzymes. So high kinetic energy, causing vibrations of amino acids, breaking hydrogen and ionic bonds in the tertiary structure of this time a named enzyme Rubisco, resulting in a change in its active site structure. So it's no longer complementary to the substrates, but I'm naming the substrates now as RuBP and carbon dioxide. They can't react to form GP, so there's going to be less GP, less TP, less RuBP recycled, it said at the end over there. So again, there's enough detail to get the 2 marks. Okay, last application. Okay, so we are going to now move on to the final application of limiting factors of photosynthesis, which is a farming application. So here I'm asking you to suggest, I've not told you, how a commercial grower could utilise his knowledge or her knowledge of limiting factors to increase crop yield within a glasshouse. Okay. Then I'd like you to think about this commercial grower. If they're considering increasing temperature in the glasshouse during winter to 25 degrees C, what should the grower need, what do they need to consider before doing that? And then finally, just a little thinking point for essays really. Global warming, will this be an advantage to growers or not? Have a go, write your ideas down and then rejoin me. Okay, so if you are a commercial grower and you know about the limiting factors of photosynthesis, then you can ensure they are not limiting. Okay, so how you might apply this is to your glasshouse design. So you can have lighting fitted, so that in winter or when light intensity does drop, with that being such an important limiting factor, you can switch on lights and illuminate your crop. Also, it's possible to pump carbon dioxide into your glasshouse to control your carbon dioxide temperature. Your carbon dioxide concentration, and you can also control your temperature and keep it close to the optimum temperature by having windows for ventilation to let heat out on hot days, but also adding heaters to heat your crop so you can keep the optimum temperature within your glasshouse. Okay. So quite often the data you might get might relate to limiting factors, studies of the limiting factors of different crops, because obviously the grower needs to know the optimum temperature, the optimum light intensity and carbon dioxide concentration for the crop species. A really common question though that they always ask in these farming questions is this one here. So it's this idea: in winter you might want to heat your glasshouse, but what do you need to consider at the same time? Well, if you're in winter, then the light intensity is going to be really low. And if the light intensity is low, there is no point increasing the temperature, because the light intensity will be the limiting factor first of all. So you need to make sure that the light intensity is optimum before you start considering increasing your temperature. There'll be a cost associated with heating your glasshouse, and it would be a waste of energy. But even worse than that, if you were to heat your crop under those conditions, you could cause an increase in respiration rate of your crop. So because respiration obviously is an enzyme catalyzed reaction as well, but it's not limited by light, so you could increase the rate of respiration, and that could lead to the plants using glucose up that would then limit the growth of the plant. So that is an important consideration. Okay, so if light were limiting, increasing the temperature wouldn't give you an increase in rate of photosynthesis, but it might increase your rate of respiration. Well, it would increase your rate of respiration and would actually cause a reduction in yield. So you're actually losing money to lose money further. Okay. Finally, global warming. So we know that the planet is warming, and some people have suggested this might be an advantage to certain growers. It kind of depends where you live in the world. So if you're quite close to the optimum temperature for your crop species already, then that will not be an advantage to you when the temperature goes up, because you might exceed the optimum. It obviously affects other things, like where I'm thinking my geographers now will be thinking about the impact on weather systems, and things like water availability. Of course, water availability doesn't directly limit photosynthesis, but if you're in dry conditions, the plant will close the stomata, and if the stomata are closed, then carbon dioxide can't get into the plant for photosynthesis, so that would limit the photosynthesis rate. Okay, so we're moving on from photosynthesis now to energy transfer next week. So the final thing, just to see whether you've got any of that knowledge left in your brain, just write anything you know now about energy within organisms and energy transfer, and we will recap it in our remote. Okay, right, thank you very much.